Hi, everyone. So our next speaker is Winston Yan, who's very anxious to get up here. He's currently graduate in the Fangzhang side, and I think Keith mentioned as well. So his work is actually characterizing regulatory as a pastime for, mostly actually, I think, for AAV delivery that we use for research application, but hopefully in the future for therapeutics. So, thank you very much for this invitation. Coming to this meetings today, so I have the pleasure of giving you this description of the step 4 Cas9. Collected the question for a great introduction, because right now, the primary Cas9 that we're using for engineering is strep pyogenes, right? And we've been remarkably lucky to see that strep pyogenes work so well out of the box, but clearly there's some limitations, both in terms of the targeting space, as well as I'll demonstrate the side is really a limitation to in vivo studies. So, what I want to tell you about is the new Cas9 we characterized from the orthologous species, Staph aureus. And with it, we also provide a framework for potentially engineering more future variants that can be engineered for targeting various. Alright, so again, this is the key of this is engineering a smaller Cas9 chip from the in vivo editor. And I think if we're thinking about drug discovery, which is the topic of this conference, there's a lot that we can figure out in cells, a lot of cell biology and a lot of screening that can happen readily, but ultimately, much of the biology has to be moved to in vivo systems if we're going to eventually turn them into therapeutics. And I mean in vivo systems not only just in terms of in vivo systems not only in terms of mouse models and such, right? But also other animals, model organisms such as rats, potentially non-human primates. And increasingly we're starting to realize that there are differences, even though mice are tremendously useful and similar, there are differences, so we need to validate them in other organisms. The other part about having the need for this generalizable, yeah, check it. Oh, there we go. That's a lot more. Alright, sorry about that. So the other reason why we need this generalizable system is that right now, when we much of the community has already generated tons and tons of genetically different models in mouse lines. And to test out all these different genotypes that are coming out of genome-wide association studies, to cross them together and to make these new mouse lines would be incredibly labor intensive. If there was some easy, versatile, and efficient way that we can deliver Cas9 to make these engineered mutations, that really gives us a big advantage in terms of throughput and speed and ultimately cost. So what I'm going to argue is that one of the best ways that we can deliver Cas9 or any other really Cas9 here today into animals will be through a viral vector called a adeno-associated virus. So I'm going to first describe to you why we're going to choose an adeno-associated virus. I'm going to tell you why that motivated us to look for a smaller Cas9. And then next, I'm going to show you a bit more about a different genome-wide off-target detection method that we've evaluated. And lastly, I'm going to show that this actually works very robustly in vivo in mice. So adeno-associated virus has been attractive in the gene therapy field for a number of years, and has many of these advantages that you probably are familiar with. So you have low immunogenicity, low oncogenic potential, and perhaps importantly, you have a variety of different serotypes that you can target to different organs, and these are still being evolved today. So every couple of months, new serotypes emerge that either have more organs specificity or potentially more activity in those target organs. And very importantly, if we're thinking about drug discovery, is that there is a therapeutic precedent. So the therapeutic precedent comes from there are existing therapies just for delivery of a transgene using adeno-associated virus, and that provides a nice platform to build on if you are to carry Cas9 into more therapeutic contexts. So if we're trying to put something inside AAV, where do we go with the existing Cas9 system? So the existing Cas9 is strep pyogenes Cas9, and what we have is this large transgene here. So the work that's been done in our lab by two of the postdocs have described, they've really done some nice work using a two-vector system for delivering strep pyogenes Cas9, where one contains a promoter driving the SP Cas9 and another contains the guide, an array of different guides. And they've used this to demonstrate some nice biology. This is the publication here. But the limitations of that are one, that the promoter choice is limited. Even when you have one system here, they use a 235 base pair promoter and a very short poly A sequence. And even that, they're really pushing the size limits of it. So a lot of promoters, when we're trying to have organ specificity or just to generally allow versatility, they don't exist in the size range. They're more in the 400, 600, 1,000 base pair range. Secondly, when we think about your co-transduction efficiency, in organs that are hard to transduce, you might have a problem where co-transducing those into the same cell to deliver both these necessary components together might be low. So imagine if there's 40% efficiency and there's 40% efficiency, and you combine that, you actually get less than 20% efficiency. So anyways, these are some limitations that led us to think about developing a single vector that contains both Cas9 and the guide RNA. So again, this is just some detailed math of showing you why a single vector using the SP system actually wouldn't work. When you combine together this Pol3 promoter and sgRNA, so this commonly instead of U6 sgRNA, plus the Cas9 from strep pyogenes and the poly A tail, together you're already at the packaging limit of AAV. So that's a big problem in the gene engineering field, where the AAV packaging limit is often, it's a very nice vector, but the packaging limit is tough for a lot of applications. So what do we do to search for this smaller Cas9? This is work that I did together with Anne and Love from our lab. They're through the pioneers in engineering Cas9, and a lot of the early publications bear their name. So what we first did was we did a metagenomic screen to search this space of type 2 CRISPR systems looking for smaller Cas9s. Next, we took those Cas9s and characterized them in vitro. We looked to see if they can actually cut and how we can reconstitute all the various components together. After that, with one of the ones, which Cas9 had in the bag, Staph aureus, we were able to produce AAV with it. And lastly, we showed that there was, we were able to validate the in vitro, or in vivo. So, remember that CRISPR is not just from one bacteria. In fact, CRISPR is a huge family of organisms. I think over 40% of bacteria and archaea contain CRISPR systems. So in particular, we're interested in the type 2 system, the details of which aren't exactly important, but these type 2 systems contain this one big endonuclease, the Cas9 protein. And Cas9s actually exist in a number of different sizes here. So the strep pyogenes Cas9 that we know and love comes around here, around over 1,300 amino acids, where there's a smaller ones from strep thermophilus, and strep thermophilus one comes in at just 1,100 amino acids. So we could engineer the other ones to use, and people have. So our lab and a number of other labs have demonstrated the usefulness that this does work, but the usefulness is a bit more limited because this has a very restricted PAM. Additionally, when we tried experiments to truncate strep pyogenes Cas9, it didn't work as well. So basically, either you're left with a more limited space which you can target, or you can have something that would have reduced activity. The fact that there are more smaller Cas9s that exist in the space, though, meant that there's more options to explore. So what we did was, with that as a design criteria in mind, we took strep pyogenes and thermophilus and compared them against smaller Cas9s here. And those 6 smaller Cas9s that we characterized, we first had to reconstitute the whole system. Remember that it's not just something that we can just take a Cas9 and use it with the existing guides. We have to reconstitute the tracer and the CRISPR RNA, which together make what we know as the sgRNA. And furthermore, we have to discover the PAM for these new sequences as well. So the details of this are actually, I'll spare you today since this is more about the in vivo part of it. when we reconstituted these systems, we found that most of them actually worked in a cell lysate assay, where we express the Cas9 in a hex cell, and then we just put that cell lysate together with the guide RNA targeting a template that we deliver. And most of these actually do cut. You can see that there are these cutting bands down here, and of course strep pyogenes is the most optimized and also the strongest. But when we switch over to actual in vivo inside a cell, so a different form of in vivo, when we switch over to that, only very few of them cut. So only the ones from Staph aureus really showed any sort of similar activity. So that's something that we're still trying to figure out why it's the case. But we were lucky enough to end up with one of these candidates that we can proceed into the next steps of our analysis. So in the next steps of in vitro characterization, we saw that it wasn't just one target that it was able to cleave. Staph aureus Cas9 was actually able to do it the same thing over a multitude of targets, and to do that with a similar efficiency to strep pyogenes Cas9. Secondly, we know that we were able to discover the PAM, and this NNGRT PAM is actually slightly more limited in targeting space, but overall much better than the orthologs that were previously found in the same size range. So we thought this was a workable system that we could proceed further. Okay, so now specificity is obviously a big concern, and Keith gave a great primer onto why we want to have an unbiased genome-wide specificity system. His GuideSeq is really a great study and does a comprehensive characterization of a lot of different targets. What we wanted to do is use a similar unbiased method to look at this comparison between SA and SP specifically. So this method is called BLESS, and I'll spare you going through this acronym, but in essence what it is that you first just cut it. So you have the Cas9, cut the DNA. You tag it. So on the cut pieces of DNA, you can actually ligate on biotinylated adapters. And then you grab it. So using the biotin, you can actually pull down those fragments of DNA. You shear the DNA to make these small, having another primer and another primer here on the biotinylated bead, and you can amplify and enrich that for a sequencing library that now contains your double-stranded break fragments, which are enriched. So, and hopefully there's going to be a question of this versus GuideSeq, and I think it's not really fair to put them in head-to-head with each other, because I think this is a complementary method. This is something that looks directly at the cuts rather than any downstream integration site, and potentially that's just as useful in vivo applications where you might not be able to introduce those modified tags, or similarly, it might be useful in situations where those tags might cause it's difficult to deliver into the cell. So one of the challenges with this is data analysis, and you can see that these are plots that show where these reads map. These reads from these sequencing reads from this experiment, and when you map them, it turns out that there's a lot of DNA hotspots across the genome. But we can differentiate those from our actual Cas9 cleaved sites, because the Cas9 cleavage events are actually very sharp. And so when you plot a histogram of the distances between all these reads, what we call this pairwise distance, we can see that for the noise, it's really a broad peak, whereas it's much sharper and asymmetric for Cas9. So that's how we were able to separate out the signal from the noise. So in doing so, we were able to count up the number of reads at a given loci, and we can produce graphs like this, where this is essentially the strength of cutting, and this is across the chromosome. So it's sort of a Manhattan plot, if you will, except of the double-stranded breaks. So this is SA Cas9, this is SP Cas9, we were able to find that there are, I think it's too early to say whether anyone is more specific or not, but at least they're comparable in specificity. And what the experiment that we did was we can actually nest inside a guide for SP and SA, so it has the exact same target, and we use that to directly compare the efficacies. So in doing so, we found that they're at least as good as the specificity of SA is at least as good as SP. And to reiterate some of the points that Keith hit home, this is a graph showing the different targets that we found, the off-target sites. When we looked at the off-target sites, we actually were able to see that first, CHIP is really not a good, it's almost an orthogonal space of finding these off-targets. Also, the current methods of predicting the off-target based on similarity, they missed a couple of these that had these bulges in the sequence. And lastly, you can see about the strength of the green color here that it really is, there's a huge false positive rate happening just as Keith described. So switching gears a little bit, we'll go to the final part, which is the actual AAV production and in vivo validation. So when we go into AAV using SA-Cas9, the math here works out a lot better. We have, this is a single vector that contains both the SA-Cas9 and its guide RNA, and together it adds up to only 4 kb. So that leaves about 600 base pairs left for promoters, which easily fits many generalizable promoters such as CMV, TBG, or synapsin for your various applications. So we were interested in targeting the liver, and what we did first was an in vitro validation in HEPA cells. We produced the virus after we found the targets that were the best in the HEPA cells. We injected them into the mouse, and then I'll show you the downstream tissue analysis that happened. The target that we chose was PCSK9. So some of you might be familiar with this, but it's supposed to be a target that came out of human genetics as a knockdown protective, where if you actually knock down this PCSK9 gene, it reduces your risk. It's supposed to, I should say that it's associated with a lower risk of adverse cardiovascular events. So in the world, there are actually people not walking around with mutated copies of PCSK9 who are totally fine, except they have a lower risk of heart disease. So this is an intriguing target from a drug development perspective, and we thought to use it as a proof of concept for our in vivo Cas9 delivery. So here we have a time course of the indels that we found in just bulk liver tissue. So we just mashed up liver tissue and looked at our indels that were created. And we were actually able to see around 50% gene editing on just 1 week after injection. So even though we have gene editing, we'd like to see that where do we go in terms of the actual protein knockout? So we see that versus the control that was injected with just an sgRNA targeting a different locus, we have a significant decrease of the serum levels of this PCSK9 protein. Now that translates into actually a significant decrease in the serum levels of the cholesterol as well. So we were able to recapitulate this phenotype that was previously shown using knockout animals just with 1 week after our AAV injection of our Cas9 system. So obviously, Cas9 delivering a nucleus into cells is challenging, or it's potentially fraught with, how should I say, there are concerns about whether it's toxic to deliver bacterial protein into cells. So here, what we did was we did a panel of liver chemistry markers, so AST, ALT, albumin, and total bilirubin. And we found no difference between the control animals and the animals that were given GFP as just a vehicle control, as well as the animals that were given Cas9 that actually modified the locus. Additionally, on histology, we were able to see no differences as well in terms of any gross inflammatory markers. So in summary, what we did was we mined this space, a metagenomic space of Cas9 to look for a smaller Cas9 protein. We were able to find a smaller Cas9 from Staph aureus, which fit the bill in terms of being able to package into an all-in-one vector for delivery, an all-in-one AAV vector for delivery. And we demonstrated that it was highly efficient, obtaining over 50%, around 50% genome editing, even after 1 week. And this allowed us to do successful therapeutic gene editing without the apparent toxicity. So with that, I'll end, and there's a lot of people I'd like to thank, but I'd like to thank you for your attention and taking questions.
Any questions? Sure.
Great talk, Winston. So in terms of the percentage of cells that will show the knockout, what do you see would be the hurdle to achieve higher than the 50% of the knockout?
So as in the question was,
Your liver cell, for example, you achieve at A7 or whatever beyond, you have up to 50% of the cells show the knockout of PCSK9.
Yeah.
So what would be the hurdle
to go into a more complete knockout, right? Yeah, so that's a really good question, right? Because is it capped out at 50%? We haven't tried this with more genes in vivo, but perhaps some more details about what we did is informative. One, this is bulk liver tissue, right? So even though I think the 80% of the cell mass in liver is hepatocytes, they don't account for all the cells in there, and you could have other cells that are accounting for this other percentage of non-negative events. So that's even more likely because the AAV that's delivered is hepatocyte trophic. So that could be one explanation. The other explanation I'm not sure I have another theory on why it might be capping out. It could be just a transduction efficiency where the AAV at the titer that we delivered actually a fairly standard and actually fairly low on the scale of gene therapy titers. And so that might not be hitting all the hepatocytes to begin with. But definitely more testing in terms of the range of titers that you deliver as well as particular cells that you're hitting, you could make a more complete knockout, if you will.
Actually, Winston, to follow up, are there any other questions? Maybe I'll have the last question. Because the thing I noticed is that you gain 50% indels. When you look at the plasma PCSK9, you're achieving almost 100% decrease. I'm trying to understand why that would be.
Yeah, so that's a great question as well. And I think there's one, again, it could be you're knocking out significantly in hepatocytes. And so the hepatocytes are largely responsible for the secreted into the serum PCSK9. The other explanation, I've talked to Kiran Musunuru, who's an expert in this field, and they've done similar experiments with adenovirus delivery. They also saw the same phenotype of 50% knockdown and greater
She asked me the same question. I saw this data.
Yeah, so I asked him that, and he said there could be some like local homeostasis where as the production of PCSK9 is lowered, then it's maintained locally but not as secreted. But yeah, those are the biological questions actually I'm not certain about the answers to.
Oh, sorry. I guess mine wasn't the last question.
Have you continued to monitor these mice past the duration of this experiment, looked at maybe durability of the response, how stable the response is?
Continued. The ones that we harvested the livers for, unfortunately, they are no longer with us. But the ones that we just took serum from, we actually continued it out for multiple months, and they do still have that same response. In terms of gross phenotype, they look healthy, they're getting pretty chubby, they're walking around. I mean, we haven't done the full necropsy to look for if there are any off-target effects or anything different that has come out. But that's definitely something that needs to be done at a larger scale to not just with the 15 mice in this cohort, but really 100, 150 mice taken at different time points to understand that more.
And the cholesterol lowering is still there?
Yes, the cholesterol. So we've had some mice we've had, I think it was 7 months afterwards, the cholesterol lowering effect is still there. Yeah.
Great. Thank you, Winston.
Well, I should add that these things the plasmas are all available at Gene as well, and I'm happy to take any questions there. So thanks.